Hi guys, so this is a recorded lecture on Chapter 12 of your Cohen text. As you recall, we had a little bit of an introduction to 12.1 in the previous lecture. So we'll start off with section 12.2, talking about interactions between the drug and microbe. So one important thing to think about when it comes to choosing a drug and the microbe that you're trying to kill off is this concept of selective toxicity. So this is a central concept in antibiotic treatment. So ideally, the antimicrobial drug that's chosen to kill off the microbial cells causing disease should either kill or inhibit the cells and at the same time minimize damage to the host tissues. So obviously there are many drugs out there, but the best drugs that are currently used are either going to block the actions or synthesis of molecules or chemicals or structures in microorganisms, but they're not going to hurt the vertebrate cells. And hopefully some of the things we talked about in Chapter 4 and Chapter 5 when we learned about different things that can be found in microorganisms but are not found in vertebrate or eukaryote cells, hopefully those things will they'll come back to mind when we discuss this. So some of the drugs that have just really excellent selective toxicity are the penicillins. Obviously, these are one of the first drugs available and used as an antibiotic. So their mode of action is to block the synthesis of the cell wall. And of course, this is found only in bacteria. So if you have a bacterial target, then this is going to hurt and kill the bacterial cells, but not the human cells. Some of the drugs out there are toxic to human cells. And that is because drugs that act upon a structure that's found in both the infective agent and the host cell are, targeted with this toxic drug. So not only is it going to be hurtful to the bacterial cells, it's also going to hurt the human cells. So as the characteristics of the infectious agent become more similar to that of the host, such as in helminths, which are multicellular and are eukaryotic, the selective toxicity that one would ideally have becomes more difficult as the characteristics of that parasite becomes more like the host it's infecting. All right, so mechanisms of drug action. So we're going to go through these individually, but these are some of the metabolic targets that are chosen that the chemicals are going to act upon. And a lot of times it's inhibition, either destruction of or doesn't allow for the production of certain molecules. So the first one, which we talked about with penicillins, is inhibition of cell wall synthesis. So as you recall, peptidoglycan is a main component of that cell wall, and so in this case, we're going to inhibit that peptidoglycan synthesis, or we're going to break down the wall that's already intact. Another one is inhibition of the nucleic acids, such as the DNA or RNA, in both their structure and function. Function, again, being things like replication and transcription. Inhibition of protein synthesis. One thing we learned that was different in regards to eukaryotes versus prokaryotes was the size of the different ribosomal RNA subunits. So that whole idea will come back into play when we talk about how these drugs can inhibit protein synthesis. And the last one is interference with the cell membrane structure or function. This one can be a little bit more difficult. There's more selective problems with selective toxicity. A lot of times because the cell membrane structure and function is similar in both prokaryotes and eukaryotes. And the last one we'll talk about is inhibition of folic acid synthesis. So this is a good picture that kind of just goes through and overviews kind of everything we'll talk about in regards to mechanism of drug action. So the first one we'll talk about is over here. This little pink layer is what they're demonstrating as being a cell wall in this microbe. So there's going to be a whole list of different cell wall inhibitor drugs. And again, their mode of action is either to block the synthesis or repair to the cell wall. And the most common one is penicillin. That we'll also talk about all these other common cell wall inhibitors. The next one on the list, I believe, will be DNA and RNA. So if you recall, this is the whole process of where this is the plasmid. And in this case, the arrow here is Xing out the DNA replication and transcription, as well as in some cases the messenger RNA will get transcribed. And in this case, there's certain drugs that will actually inhibit the RNA polymerase. Another main target is the protein synthesis inhibitors acting on the ribosomes. Again, in prokaryotes, there is the 50S and 30S ribosomal subunits. These are found in prokaryotic cells. Eukaryotic cells have a 60S and a 40S subunit. So this is one way that these drugs can be selectively toxic because they're only interfering with the bacterial ribosomal subunits. And so we'll talk about a few that work specifically on the 50S subunit, some that work on the 30S subunit, and then some that work and block both the 30S and the 50S. As well, this is sort of the next one we'll talk about with the cell membrane. There's only a couple of classes of or types of molecules we'll talk about that are in this category, but essentially they'll target the cell membrane, causing loss of selective permeability, and probably the one we'll talk about most is the polymyxins. And then our last class is the folic acid synthesis in the cytoplasm. In this case, there's a couple of types of drugs that are going to block the pathway and inhibit metabolism.